Good morning, everyone. The chapter that I am going to read today is Fermentation. Fermentation is a metabolic process that converts sugar to acids, gases, and alcohols. It occurs in yeast and bacteria, and also in oxygen starved muscle cells. As in the case of lactic acid fermentation, fermentation is also occurred more broadly to refer to the bulk growth of microorganisms on the growth medium, often with the goals of producing a specific chemical products like enzyme, vaccines, antibiotics, food product or additives, etc. French microbiologist Louis Pasteur is often remembered for his insights into fermentation and its microbial causes. The science of fermentation is known as zymology. Fermentation takes place in the lack of oxygen when the electron transport chain is unusable and becomes the cell's primary means of ATP production. It turns NADH and pyruvate produced in the glycolysis step into NAD positive and various small molecules depending on the type of fermentation. In the process of O2, NADH and pyruvate are used to generate ATP in respiration. This is called oxidative phosphorylation and it generates much more ATP than glycolysis alone. For that reason, cells generally benefit from avoiding fermentation when oxygen is available, the exception being obligate anaerobics which cannot tolerate oxygen. The first step, glycolysis, is common to all fermentation pathways, that is C6H12O6 plus 2 NAD positive plus 2 ATP plus 2 inorganic phosphate is equal to 2CH3CO COO negative plus 2 NADH plus 2 ATP plus 2 H2O plus 2 H positive. Pyruvate is CH3CO, COO negative. PI is inorganic phosphate. 2 ATP molecules and 2 PI are converted to 2 ATP and 2 water molecules via substrate level phosphorylation. 2 molecules of NAD positive are also reduced to NADH in oxidative phosphorylation. The energy of ATP formation is derived from the electrochemical proton gradient generated across the inner mitochondrial membrane, or in the case of bacteria, the plasma membrane, via the electron transport chain. Glycolysis has substrate level phosphorylation, ATP generated directly at the point of reaction. Humans have used fermentation to produce food and beverages since the Neolithic age. For example, fermentation is used for preservation or in a process that produces lactic acid as found in such sour foods as pickled cucumbers, kimchi, and yogurt, as well as for producing alcoholic beverages such as wine and beer. Fermentation can even occur within the stomachs of animals such as humans. Definitions of fermentation. Too many people fermentation simply means the production of alcohol. Grains and fruits are fermented to produce beer and wine. If a food sold, one might say it was off or fermented. Here are some definitions of fermentation. They range from informal general uses to more scientific definitions. First, preservation methods for food via microorganisms. Second, any process that produces alcoholic beverages or acidic dairy products. Third, any large-scale microbial process occurring with or without air. 4. Any energy-releasing metabolic process that takes place only under anaerobic conditions. Fifth, any metabolic process that releases energy from a sugar or other organic molecules, does not require oxygen or an electron transport chain, and uses an organic molecule as the final electron acceptor. Examples of fermentation. Fermentation does not necessarily have to be carried out in an anaerobic environment. For example, even in the presence of abundant oxygen, yeast cells greatly prefer fermentation to aerobic respiration. As long as sugars are readily available for consumption, a phenomenon known as the Crabtree effect. The antibiotic activities of hops also inhibits aerobic metabolism in yeast. Fermentation reacts NADH with an endogenous organic electron acceptor. Usually this is pyruvate formed from the sugar during the glycolysis steps. During fermentation, pyruvate is metabolized to various compounds through several processes. First, ethanol fermentation, aka alcoholic fermentation. This is the process of ethanol and carbon dioxide. Second, lactic acid fermentation refers to two means of producing lactic acid. First, homolactic acid fermentation is the production of lactic acid exclusively. Second, heterolactic fermentation is the production of lactic acid as well as other acids and alcohols. Sugars are the most common substrate of fermentation, and the typical examples of fermentation products are ethanol, lactic acid, carbon dioxide, and hydrogen gas. However, more exotic compounds can be produced by fermentation, such as butyric acid and acetone. Yeast carries out fermentations in the production of ethanol in beers, wines, and other alcoholic drinks, along with the production of large quantities of carbon dioxide. Fermentation occurs in the mammalian muscles during periods of intense exercise where oxygen supplies become limited, resulting in the creation of lactic acid. Ethanol fermentation. The chemical equation below shows the alcoholic fermentation of glucose, whose chemical formula is C6H12O6. One glucose molecule is converted into two ethanol molecules and two carbon dioxide molecules. C6H12O6 is equal to 2 ethanol plus 2 carbon dioxide. C Before fermentation takes place, one glucose molecule is broken down into two pyruvate molecules. This is known as glycolysis. Lactic acid formation. Homolactic fermentation producing only lactic acid is the simplest type of fermentation. The pyruvate of glycolysis undergoes a simple redox reaction forming lactic acid. It is a unique because it is one of the only respiration processes to not produce a gas as a byproduct. Overall, one molecule of glucose or any 6-carbon sugar is converted to 2 molecules of lactic acid. It occurs in the muscles of animals when they need energy faster than the blood can supply oxygen. It also occurs in some kinds of bacteria such as lactobacilli and some fungi. It is this type of bacteria that converts lactose into lactic acid in yogurt, giving it a sour taste. These lactic acid bacteria can carry out either homolactic lactic fermentation where the end product is mostly lactic acid, or heterolactic fermentation where some lactate is further metabolized and results in ethanol and carbon dioxide via a phosphoketolase pathway, acetate, or other metabolic products. For example, glucose is converted into CH3CHO plus C2H5OH plus CO2. If lactose is fermented, as in yogurts and cheeses, it is first converted into glucose and galactose, both 6-carbon sugars with the same atomic formula. C12H22O11 plus H2 is equal to 2C6H12O6. Heterolactic fermentation is in the sense intermediate between lactic acid fermentation and other types. For example, alcoholic fermentation. The reason to go further and convert lactic acid into anything else are: first, the acidity of lactic acid impedes biological processes. This can be beneficial to the fermenting organisms as it derives out the competitors who are unadapted by the acidity, as the results of the food will have a longer shelf life. Part of the reason foods are purposely fermented in the first place. However, beyond a certain point, the acidity starts affecting the organisms that produces it. The high concentration of lactic acid, the final product of fermentation, derives the equilibrium backwards, Le-Chatelier's principle, decreasing the rate at which fermentation can occur and slowing down growth. Ethanol that lactic acid can be easily converted to is volatile and will readily escape, allowing the reaction to produce easily. Carbon dioxide is also produced, however, it's only weakly acidic and even more volatile than ethanol. Acetic acid, another conversion product, is acidic and not as volatile as ethanol. However, in the presence of limited oxygen, its creation from lactic acid releases a lot of additional energy. It is a lighter molecule than lactic acid, then forms fewer hydrogen bonds with its surrounding due to having fewer groups that can form such bonds, and thus more volatile it and will also allow the reaction to move forward more quickly. If propionic acid, butyric acid, and longer monocarboxylic acids are produced, the amount of acidity produced per glucose consumed will decrease, as with ethanol, allowing faster growth. Aerobic respiration. In aerobic respiration, the pyruvate produced by glycolysis is oxidized completely, generating additional ATP and NADH in the citric acid cycle and by oxidative phosphorylation. However, this can occur only in the presence of oxygen. Oxygen is a toxic to organisms that are obligate anaerobes and is not required to facultative anaerobic organisms. In the absence of oxygen, one of the fermentation pathways occurs in order to regenerate NAD positive. Lactic acid fermentation is one of these pathways. Hydrogen gas production in fermentation. Hydrogen gas is produced in many types of fermentation: mixed acid fermentation, butyric acid fermentation, caproate fermentation, butanol fermentation, glyoxylate fermentation as a way to regenerate NAD positive from NADH. Electrons are transferred to ferredoxin, which in turn is oxidized by hydrogenases, producing H2. Hydrogen gas is a substrate for methanogens and sulfate. reducers, which keep on concentration of hydrogen low and favor the production of such an energy rich compounds, but hydrogen gas at a fairly high concentration can nevertheless be formed as in floats. As the example of mixed acid fermentation, bacteria such as Clostridium pasturionum ferment glucose producing butyrate, acetate, carbon dioxide and hydrogen gas. The reaction leading to the acetate is C6H12O6 plus 4H2O is equal to 2CH3COO negative plus 2HCO3 negative plus 4H positive plus 4H2. Glucose could theoretically be converted into just carbon dioxide and hydrogen, but the global reaction releases little energy. Methane gas production in fermentation. Acetic acid can also undergo a dismutation reaction to produce methane and carbon dioxide. CH3CO negative plus H positive is equal to CH4 plus CO2. Delta G naught is equal to minus 36 kilojoule per reaction. The disproportionation reaction is catalyzed by methanogen archaea in their fermentative metabolism. One electron is transferred from the carbonyl function electron donor of the carboxyl group of the methyl group electron acceptor of the acetic acid to respectively produce carbon dioxide and methane gas. History of fermentation. The use of fermentation, particularly for beverages, has existed since the Neolithic and has been documented dating from 7000 to 6600 BCE in Jehu, China, 6000 BCE in Georgia, 3150 BCE in ancient Egypt, 3000 BCE in Babylon, 2000 BCE in pre-Hispanic Mexico, and 1500 BCE in Sudan. Fermented foods have a religious significance in Judaism and Christianity. The Baltic god Rugutis was worshipped as an ancient agent of fermentation. Louis Pasteur, 1822 to 1895, during the 1850s and 1860s, showed that fermentation is initiated by living organisms in a series of investigations. In 1857, Pasteur showed that lactic acid fermentation is caused by living organisms. In 1860, he demonstrated that bacteria causing souring in milk, a process formerly thought to be merely a chemical change, and his work in identifying the role of microorganisms in food spoilage led to the process of pasteurization. In 1877, working to improve the French brewing industry, Pasteur published his famous paper on fermentation, Idées sur la bière, which was translated into English in 1879 as Studies on Fermentation. He defined fermentation incorrectly as life without air, but correctly showed that specific types of microorganisms cause specific types of fermentations and specific end products. Although showing fermentation to be the result of the action of living microorganisms was a breakthrough, it did not explain the basic nature of the fermentation process or prove that it is caused by the microorganisms that appear to be always present. Many scientists, including Pasteur, has unsuccessfully attempted to extract the fermentation enzyme from yeast. Success came in 1897 when the German chemist Edward Buchner ground up yeast, extracted a juice from them, then found to his amazement that this dead liquid would ferment a sugar solution, forming carbon dioxide and alcohol much like living yeasts. Buchner's results are considered to make the birth of biochemistry. The unorganized ferments behaved like the organized ones. From that time on, the term enzyme came to be applied to all ferments. It was then understood that fermentation is caused by enzymes that are produced by microorganisms. In 1907, Buchner won the Nobel Prize in Chemistry for his work. Advances in microbiology and fermentation technology has continued steadily up until the present. For example, in the late 1970s, it was discovered that microorganisms could be mutated with physical and chemical treatments to be higher yields, fast growing, tolerant of less oxygen, and able to use a more concentrated medium. Strain selection and hybridization developed as well, affecting most modern food fermentations. Other approach of advancing the fermentation industry has been done by companies such as Biotok, a biotechnology company that naturally involves microorganisms to improve fermentation processes. This approach differs from the more popular genetic modification which has been currently used in the industry standard. Industrial fermentation. Industrial fermentation is the intentional use of fermentation by microorganisms such as bacteria and fungi to make products useful to humans. Fermented products have applications as food as well as in general industry. Some commodity chemicals such as acetic acid, citric acid, and ethanol are made by fermentation. The rate of fermentation depends upon the concentration of microorganisms, cells, cellular components, and enzymes, as well as temperature, pH, and for aerobic fermentation, oxygen. Products recovery frequently involves the concentration of the dilute solution. Nearly all commercially produced enzymes such as lipase, invertase, and rennet are made by fermentation with genetically modified microbes. In such cases, production of biomass itself is the objective. As in the case of baker's yeast and lactic acid, bacteria starters, cultures for cheese making, in general, fermentations can be divided into four types: production of biomass, production of extracellular metabolites, production of intracellular components, transformation of substrate. These types are not necessarily disjoint from each other but provide a framework for understanding the differences in approach. The organisms used may be bacteria, yeast, molds, animal cells, or plant cells. Special considerations are required for the specific organisms used in the fermentation, such as the dissolved oxygen levels, nutrient levels, and temperature. General overview of industrial fermentation. In most industrial fermentations, the organisms are submerged in a liquid medium. In others, such as the fermentation of cocoa beans, coffee, cherries, in miso, fermentation takes place on the moist surface of the medium. There are also industrial considerations related to the fermentation processes. For instance, to avoid biological process contamination, the fermentation medium, air, and equipment are sterilized. Foam control can be achieved by either mechanical foam destruction or chemical anti-foaming agents. Several other factors must be measured and controlled, such as pressure, temperature, agitators, shaft power, and an important element for industrial fermentations is scale up. This is the conversion of a laboratory procedure to an industrial process. It is well established in the field of industrial microbiology that what works well at the laboratory scale may work poorly or not at all when first attempted at large scale. It is generally not possible to take fermentation conditions that have worked in the laboratory and blindly apply them to the industrial scale equipment. Although many parameters have been tested for use of scale up criteria, there is no general formula because of the variation in the fermentation processes. The most important methods are the maintenance of constant power consumption per unit of broth and the maintenance of the constant volumetric transfer rate. Thank you.